Go. All right, so we made it to the end of the week, folks, and I'm hoping that you're all excited. I'm joined here by my friend, my good friend. Her name is Bordy, so we're having our own celebration here. She's trying to escape from me, and I hope that you're not trying to escape from physiology. We're almost at the end of this week's lectures. It's a long haul, hoping that you're all in a good mood. My dog is in a great mood, so I'm gonna let her go and grab the snack that she wants, and we'll get going with our lecture, but we made it. All right. Okay. All right. And by the end of today's lecture, one of the things that I hope you will understand, if you can see it, is why I have this. And this is actually a pineapple, as you can see. And if you're wondering why I'm holding a pineapple and why this 3-pound pineapple is so important to our lecture, you're just going to have to wait until I'm done. But we'll get back to this a little bit later on. All right, so why don't we just go ahead, jump right in to the lecture material that we have, and I'll start sharing my slides, and we can get going into lecture number 6, where we're talking, as you can see, about floating brains. And floating brains in physiology is actually a good thing, and we'll talk a little bit more about it. I know that you've been listening to these lectures so far, but I haven't heard from you yet. I hope that you will connect with me when you can. Email, Microsoft Office Teams, discussions, the Collaborate session on Thursday, whenever it works best for you. I'm kind of looking forward to kind of meeting you all. So hopefully that'll all work out. So let's just jump right into it. So today we're going to be talking about the blood-brain barrier. It's a really, really interesting structure that actually protects the brain. We'll talk also about astrocytes and how astrocytes are critical to the maintenance of things that happen within the brain, especially on regulating blood flow, and that mechanism is one that, hint, hint, you need to know. So we'll get to that in a few minutes. And then we'll also be talking about where the brain is actually floating in, and it's floating in CSF, and what that CSF is actually doing, and how it's getting formed by a specialized structure within the ventricles called the choroid plexus. All right, so off we go into our last lecture of the week. So the blood-brain barrier is actually a really important part of the nervous system. So the brain and the spinal cord are both protected from all of the different substances, including drugs and ions that are found in the general circulation, so the blood, and from the rest of the body. And it's because the ionic composition of the extracellular fluid, the fluid that's bathing all of the neurons, has to be very, very tightly regulated or very carefully controlled. We can't alter the excitability of the membrane. So remember, we were talking about potassium chloride, and we're talking about if we increase the amount of potassium outside of the cell, it would actually cause depolarization. And if we had too much depolarization, because of the way in which these voltage-gated channels inactivate, if they're continually depolarized, it's actually going to cause a depolarization block, and so no more action potentials are going to be produced. We also can't have neurotransmitters floating around, so we need to tightly regulate that. So that's also a concern. So the blood-brain barrier is important to regulate anything that's produced outside of the brain. And in fact, we know now in 2020 that if you look at different parts of the body, including what we call the gut microbiome, the bacteria in your gut actually produce a lot of neurotransmitters, and they produce neurotransmitters like serotonin at levels that are even higher than we would find in the brain. And we need to keep that out of the brain because we need that just from neurotransmission. So this extracellular fluid has to be very carefully maintained. It's really important. And the way that we can control that and control what gets into the brain and what ions can flow into the brain is through this structure known as the blood-brain barrier. So we can think of this blood-brain barrier kind of having a twofold function. So it can act between the blood vessels, which you see up here, and you can see the concentration of the different ions up here within the blood vessels and the capillaries, so they would be flowing through. And we have like high sodium outside, potassium levels, and of course proteins. And then we also have like this interstitial fluid. We also have this other compartment where we have to very tightly regulate this cerebrospinal fluid, or CSF, as well. And so all of these different compartments have to come into play. And of course, we have exchange between this interstitial fluid and the CSF. And these two compartments are very important, and they're found in slightly different places. So we have fluid bathing the neurons, so the fluid that's outside of the neurons themselves. And then we have the CSF, which is actually filling these empty holes within the brain that we call the ventricles. So these two different elements are actually really important. The interstitial fluid is going to interact with the fluid that is found inside the ventricles or the CSF, but we very tightly regulate what gets in and out of these compartments through the blood-brain barrier at the level of the capillaries. So this structure here, between the capillaries, which are common to both of these other compartments, common between this compartment and this compartment, these blood capillaries and controlling the flow across these blood capillaries, I hope that you can see on this diagram, is going to be really important. And in fact, we can take a look at the brain as a whole. So these blood capillaries, what makes those so special within the brain? So the majority of the brain is actually protected by very specialized blood vessels or blood capillaries, but not all of the brain is. In some places, we actually need some communication with the blood vessels, and we need a little bit of leakiness. And this is incredibly essential for neurons to be able to communicate freely with the bloodstream in certain instances. One place in particular that we'll get back to toward the very end of the lectures on the nervous system is known as the hypothalamus. And this area, the hypothalamus, it needs to be able to sense the different types of compounds and components that are in the blood vessels and then in the general circulation, because it is a very important regulator of hormonal release. And associated with that, we also have the pituitary gland, which is directly connected to the hypothalamus, which also releases hormones. And so at this area, in this junction of the hypothalamus and the pituitary, we need a little bit of leakiness. The blood-brain barrier is actually not going to be continuous there, and it's purposely a little bit leaky to allow the release of different hormones. In the circumventricular organs, so in the spaces around the third ventricle, the blood-brain barrier is also broken so that neurons can specifically sense different chemical concentrations. So when we look at it as a whole, talking about both the hypothalamus, pituitary, and in the circumventricular organs, near these ventricles, in the third ventricle that I'll get to in a moment, this blood-brain barrier is not going to function in the same way that I'm going to describe to you in the next few slides. And we need that for endocrine release. We also need it for sensitivity to plasma metabolite levels. All right, so I'm hoping that's okay. And again, we'll come back to this in a few minutes. So before we get to the ventricles, before we get to CSF, let's talk about other ways in which we can actually protect the brain. And of course, we have the skull, the outermost covering, and then we have a series of different types of membranes, and these are known as the meninges, and these are sitting under the skull on top of the brain, if you want to think of it that way. So you can actually see this diagram here. The cranium would represent the skull, and then underneath that we have these different types of membranes like the meninges. We would also have the arachnoid membrane, and then we would have here we would have the pia mater. So the dura mater, the one that's right under the skull, is a very tough membranous sac that connects the brain and the spinal cord together. Under that is this arachnoid membrane. It's not quite as tough, and if you've ever taken an anatomy lab or you've taken a neuroanatomy lab, you'll know that this dura mater is actually quite thick, quite dense, and it is pretty tough. The membrane right under that, which is the arachnoid membrane, not quite as tough, and it's much more of a delicate type of tissue. And then we have the pia mater, which sits right on top of the brain itself. So that's sort of the last layer. And it's tethered to the arachnoid by these series of trabecula that you see here. And that tethers it or connects this pia mater with the arachnoid membrane right above it. If you look very carefully on this slide, and highlighted for you again here, we also have this subarachnoid space. And this subarachnoid space is important because this space between the physical brain itself between the arachnoid membrane and the pia mater, this space that exists, this arachnoid space, the subarachnoid space, is actually important because it actually fills with CSF. And why that's important is that the fluid that fills this subarachnoid space allows for your brain, because it is found in there, it allows for the brain to float. And one of the reasons why I spoke about this a little earlier on, in this pineapple, bought specifically for this class, for the end celebration of week 1, is 3 pounds. So if you ever pick up a 3-pound pineapple, you'll be surprised at how heavy it is. And a lot of times we kind of think of the brain as being this really light structure, but in fact, the brain is about as heavy as this pineapple. So it's about 3 pounds. You don't really feel that you're supporting like 3 pounds on top of your head. Part of it is because it's actually floating in this liquid. So if I could put that in water, it would actually take a little bit of the strain of having to lift it. And that's exactly the purpose of the subarachnoid space and the CSF that's found within it. Okay, so let's jump back to the blood-brain barrier. And that's what we actually began with before this little detour. So this blood-brain barrier really consists of those specialized blood vessels. So remember that pie chart that we were looking at earlier, and the blood vessels were really important. And what makes this really special within the brain, the blood vessels themselves are a little bit different than we would find elsewhere in the body. And one of the things that makes this unique is that normally when we look at blood vessels or capillaries, the smallest type of blood vessels, they have these little pores or these little openings. And you can see that a little bit more here, and different types of cells can more freely enter, and if cells can enter, so can other substances. And these large gaps allow for better communication. And in different parts of the body, that's great, because that's exactly what capillaries are supposed to do. So we call these fenestrated, and a fenestra is a window, and these openings allow for that kind of communication, which I think is pretty cool. So you also get a bonus of just learning a little French, I guess. My French not being so great. But in the brain, these endothelial cells, these capillaries, still lined by endothelial cells, they're actually much more tightly connected with each other. And we have much less of this intercellular gap, and they're much more tightly bound. And this is what makes up the blood-brain barrier. So that's why those blood vessels in the brain are so important, and they're different. And these endothelial cells being tightly knit together really makes the difference. So we've jumped through a number of different things about the blood-brain barrier. We've talked about some of the structures that physically protect and lift the brain, and we talked about things from the dura mater all the way down to the pia mater, and then the subarachnoid space and the CSF that fills it, even though we haven't talked about it, is how we can actually float the brain, and we can also protect it from mechanical stress. So imagine your brain being as heavy as this pineapple and then being shaken back and forth. That's a lot of weight to actually press up against the skull. And we know, for example, in concussion injuries, that's exactly what happens. And without the CSF, the injuries by concussions would be much, much more severe. So it does act as a cushion. It acts as a flotation fluid, and it's also physically protecting the brain as well, which again, I think is pretty cool. So one of the other really important cells, and again, we have to think about blood vessels, endothelial cells, and we've talked a lot about neurons, and we've mentioned in passing other types of glial cells. And coming back, one of those glial cells are these astrocytes. And if we take a look at those unique capillaries that make up the blood-brain barrier, lacking those fenestrations, very tightly knit together, in the brain, we also have a lot of these astrocytes that you see here. So this would represent an astrocyte. And they send one of their processes down and actually touch the blood vessel. So imagine we're cutting through the blood vessel that's coming out toward you. And so their end feet, the astrocytic end feet, actually line these blood vessels as well. And these astrocytes are not only in contact with the blood vessel, but people started to realize they're also in direct communication with neurons, which is what you see here. And this is why we think that these astrocytes actually provide a bridge between the neurons and the blood vessels. Now you kind of have to wonder, why would they do that? And what purpose would they serve in doing so? And you can actually see on this diagram, one of the key elements of this is that the blood vessels are bringing in glucose into the brain, and neurons are actually not really good users of glucose. They've been adapted for a different type of metabolic response. And one of the things that happens is that the glucose that is carried by the capillaries, these specialized capillaries, that actually can pass through the blood-brain barrier. So it's a small enough molecule, and it also has transporters that allow it to go through. But the things that actually take it up are not neurons. They're actually these astrocytes, and they take up the glucose. They will use it in glycolysis, and they will produce lactate. And this lactate is actually important for neurons to use for metabolism. So I think, again, pretty cool adaptation for how the shift from glucose using to lactate using and providing that to neurons is a cool adaptation. And if you take a look at astrocytic stains, you can actually see them lining up on the surface of a capillary. So you'd have to imagine these dotted lines that you see here actually represent the walls of the capillary kind of running at a slight slant. And these astrocytes not only line the blood vessels or the capillaries, and they not only provide energy substrates for neurons, they also will help to remove neurotransmitters. So on that previous diagram, one of the things that you saw was that the end feet, and actually I'm just going to go back, these end feet touch the capillary. The other process actually is at the synapse. And so we know that neurotransmitters get released here, but they can also be used in the removal of these neurotransmitters as well. So they are following, latching onto, or adhering to these blood vessels, and that's what this BV means. If you want to go back and if you're not sure what the heck is a BV, this is a blood vessel, and some end feet are latched onto the blood vessels, and others with neurons acting as that bridge that I showed you earlier on. And again, you're just seeing this in a slightly different way, and we have these really tight junctions between the endothelial cells on the particular capillaries that we see here. And you see these astrocytes in communication, one end feet from this astrocyte coming back, and then bridging with this particular neuron. All right, so another reason why they might do this is that astrocytes are in touch, or are touching the blood vessels. One of the hypotheses is that these astrocytes are sensing neuronal activity. As neurons become more active, they need more energy. And so these astrocytes might actually be regulating local blood flow. So could they potentially do that? So they're already in the right place. They're the bridge between the blood vessel and the neuron. So they're in a great spot to signal to the blood vessel when we need to bring in more nutrients to the really hardworking neurons, so they can open up, dilate, or constrict, and decrease blood flow when they constrict. And so they might have this ability to do that. And in fact, it turns out that when researchers in the 1980s started to examine if that was indeed one of their many, many roles, it turns out that they have this capacity to sense metabolic changes in neurons and to communicate that backwards to the blood vessel as well. So they're not only taking nutrients from the blood vessel, which you are seeing here. So this is a blood vessel. Here is the lumen, or the whole of the blood vessel. So we would have red blood cells, we would have glucose and everything else coming through. And we can see part of the stain of the end feet of the astrocytes that are in direct contact and that are stained with an antibody stain for astrocytic processes. So again, a really cool phenomenon. And one of the things that makes this also really unique is that we know the mechanism by which all of this occurs. So normally, I wouldn't really go out of my way to do this too often, but since we've reached lecture number 6, it's been a long week for you, I'm Sure. You got to see this face like how many times a day? I don't know, maybe 2 or 3 times a day. And I'm sure you're sick and tired of seeing me. But one of the things I'm going to give you a little hint on is that this is a good slide, and the next slide is a good slide for you to know. How do astrocytes actually signal that they should be increasing blood flow, and how does that process work? And it's a cool process, and this is a slide that you need to know. So start studying it, kind of put a circle on it, or whatever you do on an iPad, if you're taking notes on an iPad. So here's the thing. We know within the brain, and some of this should be jogging your memory, that at most synapses, about 80% of the synapses, they're excitatory. The major neurotransmitter for excitation to produce a postsynaptic potential that's excitatory or depolarizing is glutamate. And so glutamate at these synapses, and the vast majority are glutamate, 80%, triggers in the astrocyte. Again, really cool. So here's on the astrocyte, not on the neuron, but on the astrocyte, you have a metabotropic glutamate receptor that is responsive to the locally released glutamate. So it's not just going toward the postsynaptic neuron. Some of that neurotransmitter actually makes it to the astrocytes and the astrocyte end feet that surround that synapse as well. Again, really cool. Metabotropic glutamate receptor, intracellular cascades, and one of the things that happens is it will actually trigger an increase locally of calcium inside the cell, and this calcium wave, and it's actually beautiful if you look it up online. I'm sorry, I didn't think about this ahead of time, but if you look it up online, people have actually shown these calcium waves. You can actually watch excitation through calcium going across a field of astrocytes. And if I find that for the next episode that we have, I'll definitely try to show you that. So this wave of calcium crosses this one astrocyte. And one of the things that happens here at the other end foot that's touching the blood vessel, this increase in calcium is actually going to cause the breakdown of the lipid membrane of the astrocyte locally, and it's going to release arachidonic acid, and this is that AA. Apologies, you don't have that acronym here, but the AA refers to arachidonic acid, and this in turn will cause the release of prostaglandin E2, or PGE2. And this is going to be released from the end foot. Now, again, big whoop, unless you happen to be a really big fan of prostaglandin E2. Hands up if you are a fan of prostaglandin E2. So I see no one. Okay, great, awesome. So prostaglandin E2 on the blood vessel actually causes vasodilation. So it works on the smooth muscles that are normally found in the endothelial lining, and it causes their increased vasodilation. So you're going to get an increased blood flow. So every time this neuron is active, releasing neurotransmitter, so the astrocyte knows that it's active, it's going to increase the blood flow, bringing more glucose in, which can get shunted back to the neuron, which again, I think is incredibly, incredibly cool. We increase that blood flow, we increase the amount of nutrients that are available. So again, where are we sitting right now? So we know that these astrocytes are important. They are there to help to mop up any of the extra neurotransmitters, also to manage some of that extracellular potassium. They're there to provide lactate through that mechanism that I described before. We know also that there's a mechanism, and I'm winking now in case you can't see me, so winking really hard, a mechanism by which they can regulate local blood flow, and then they're also providing support for neurons through this two-way communication. So again, I think it's a really cool thing, and I hope that you also think it's a really cool thing as well. And we're almost at the end, folks, so get out whatever you're using to celebrate the end of lecture number 6 and the end of week 1. And now we're getting into some of those structures. So here are these openings or cavities that we have deep inside the brain. So here we're looking at a sagittal or a lateral view of the brain. Here we're looking at the frontal view of the brain, and we can see in gray these cavities, these openings that are going to be filled with fluid, and this is known as the lateral ventricle. And they're on the sides that you can see here, and they are paired, one on the left, one on the right. And we can see that these lateral ventricles also work from the front all the way to the back. So they take up a lot of space within the brain. This lateral ventricle will then empty into this next ventricle, which is known as the third ventricle. And you can see that here, both in cross-section, frontal view, as well as in the lateral view over here. And this, again, is this third ventricle that I spoke to you about earlier. Do you remember that third ventricle? Wasn't that long ago? Do you remember what structure or what thing that we talked about? Kind of starts with C, circumventricular organs. And again, this is an important area of the brain, and we'll come back and talk to that later. And then this third ventricle, in turn, communicates with the fourth ventricle, and this narrow little opening that you see here, and here in the lateral view, is known as the aqueduct of Sylvius that connects the third and the fourth ventricle. And so we have a way to regulate the flow of this fluid that will be found within these ventricles. So between the fourth ventricle and the central canal of the spinal cord, we will have all of these different things filled with CSF, or cerebrospinal fluid. So this again is useful for physically protecting and floating the brain. Here's a way for it to sit inside the brain, not just in that subarachnoid space that we talked about earlier, not just on the outside, but also on the inside of the brain as well. And again, these ventricles are filled with that CSF, and we talked about that at the very beginning of today's lecture. It's a bathing medium. It's a little different than the medium that we would find in the blood vessels themselves. It has very highly regulated ionic content, very few large macromolecules like proteins, etc. It's going to be produced from the blood plasma, and it's actually being produced by specialized cells called the choroid plexus. And this choroid plexus are a specialized type of cells that I'm going to show you on the next diagram that are found lining the lateral ventricle, the third and fourth ventricle, and all of these specialized cells within the third, lateral, and fourth ventricles are the ones that actually produce this fluid. And that's why these ventricles are filled with this fluid, including the subarachnoid space, because there is communication between the ventricles and the subarachnoid space. But it gets formed here at the level of the lateral, third, and fourth ventricles. All right, and eventually it doesn't just keep building up and up, because that would be bad. So remember, it's trapped inside the skull. It's now filling with fluid. We need to get rid of that fluid. And the way that this happens is that eventually the CSF will drain into the venous system and then get back into the general circulation. So this is the choroid plexus. And one of the things that makes this unique is if we are looking at the ventricle, for example, one of the things that you would see is this specialization of the cells that line the ventricle. So it's not just an empty space. There's a lining that separates the empty space from the neurons. And these types of cells are normally called ependymal cells. They normally line the ventricles, but in the lateral ventricle, the third and the fourth ventricle, these ependymal cells look a little bit different, and they look more like this structure here. And one of the things that they do is that they will actually start to make and filter some of that plasma and create the cerebrospinal fluid, which I talked about earlier. And these specialized ependymal cells are known as the choroid plexus. So eventually this fluid that gets produced is put into the ventricles, and it starts to fill the ventricles, the subarachnoid space. The CSF has the same osmolarity and sodium concentration as blood does, but it has a reduction in the levels of potassium, calcium, magnesium. It's much lower than it would be found within the blood. So that's a big difference between the blood as well as the cerebrospinal fluid. The total volume, filling the ventricles, filling the subarachnoid space, all the way down into your central canal of your spinal cord, is about 215 mils. That's not a lot. So if you look at the standard sort of can of cola, that's about 355 mils. So you take about 2/3 of that, and that's what you're going to have in terms of the fluid that's found up in your brain. The cranial CSF up in the brain itself is about 140, 25 mils in the ventricles, 115 in the subarachnoid space. CSF is about, in the spinal cord is about 75, so altogether about 215. So the vast majority is in that subarachnoid space, acting as that cushion, acting to kind of lift and float your brain. And oftentimes we want to test the cerebrospinal fluid. And what will happen is usually it's done in the lumbar region of your spinal cord, because whatever you have in your brain drains down into your central canal. It's exactly the same fluid that's produced. And hopefully you will remember that from a few slides ago. And it can be used as a diagnostic therapeutic procedure, because you can do that spinal tap. So you put a needle, you do a lumbar puncture, you get into the cerebrospinal fluid, you don't damage any neurons or any of the spinal cord, and you would draw a small sample. You're not going to take a lot. And in fact, if you drain too much through this lumbar puncture or spinal tap, the individual can actually get a headache from having too much of the cerebrospinal fluid removed. And again, in the brain, so there's just a small amount, as well as in the spinal cord itself. So we're at the end, amazingly. So we talked about the ventricles, the different structures, where CSF gets formed. And then we have looked at these specialized ependymal cells known as the choroid plexus in specific regions or specific ventricles. And then we know that it circulates around the brain and eventually will drain into the venous system. And that, I think, is about it. So we made it. Amazing. And it's Friday. Well, for me, it's Friday. I don't know when you're watching this. I'm hoping that you also are going to have amazing weather outside, and that you're also going to be able to celebrate a little bit. It's a good accomplishment, I think. You've learned some neuroscience. You get to thank your brain because it's such an amazing structure. And think about the journey that we've taken, because that was an important part of this process, that we now know a lot about neurons, action potentials, how we get depolarization blocks, how we form things like cerebrospinal fluid, and think about all the things that you've learned in this past week. I know it was a lot, but I wanted to thank you for it. So I'm just going to go ahead and stop sharing right now. I'm going to go back to this view. I'm dressed up a little bit more than I normally am because I wanted to celebrate with all of you. It's not normally how I dress at home, if you were wondering. Nor do I usually have sort of My Little Pony in the background, but hey, we made it. It's time for us to go off and celebrate. I'm going to have my favorite snack. If you don't know me very well, my favorite snack is this. I'm a very cheesy person, haha. So I'm going to go and have some of this. I hope that you're also celebrating, relaxing, having a good weekend. Thank you so much for being in this course. I haven't met any of you yet, but I hope to do so as soon as I possibly can. Hope that you get to think a little bit about neuroscience. I hope that you have a great, relaxing weekend wherever you're at, and that you continue to really love neuroscience as much as I do. Stay healthy, stay safe, and keep your synapses nice and happy, which I guess means now, after today's lecture, keep your astrocytes happy. Anyways, for another day. All right, so see you later, folks. Thanks so much. We'll see you in week number 2.